Far below shipping lanes and coastlines, a colored fragment can settle into mud deeper than any human dive. Its arrival seems impossible, yet plastic has been documented in some of the most remote deep ocean trenches. The central mystery is not whether debris can descend, but why narrow hadal valleys may retain what reaches them. Ocean trenches are not bottomless drains. They are steep, active landscapes connected to the surface by moving water. A discarded object begins among rivers, beaches, vessels, or offshore activity before currents carry it beyond familiar horizons. Sunlight, waves, and abrasion weaken larger items, producing fragments and fibers with different sizes, shapes, and densities. Some pieces float for long periods, while others sink after organisms grow upon them or sediment becomes attached. If you followed one fragment, its route would involve repeated suspension, collision, biological contact, and uncertain detours. Most plastic released near land does not travel directly to a trench, and much of its fate remains unresolved. Yet trenches offer a powerful final geometry, broad slopes descending toward narrow axes with limited escape routes. Gravity moves unstable sediment downhill, currents sweep particles along contours, and food remains fall from surface waters. Plastic can join each pathway, sometimes freely and sometimes embedded within aggregates or animal waste. The opening fragment, therefore, carries an entire transport history, even when its visible surface reveals almost none. To understand the trap, the investigation must separate measured accumulation from the tempting image of a universal abyssal landfill. Evidence grows strongest when debris identity, location, surrounding sediment, and transport setting are examined together. Oceanic plastic includes fishing gear, packaging fragments, industrial pellets, paint particles, and synthetic textile fibers. Their polymers behave differently because density, additives, weathering, and attached material influence buoyancy and degradation. Polyethylene and polypropylene commonly begin buoyant, whereas denser polymers may sink more readily in seawater. Even buoyant plastic can descend when biofilms, mineral grains, or marine snow increase the effective density. Marine snow consists of sinking biological debris, mucus, fecal material, and particles loosely bound into fragile aggregates. A small fiber caught inside an aggregate follows its descent until decomposition or turbulence releases it again. Zooplankton may ingest microplastic, then package it within fecal pellets that sink faster than isolated particles. That cue is not intentional transport, but feeding behavior can redirect plastic through the biological carbon pump. Larger carcasses and discarded feeding structures can also carry attached fragments toward the seafloor. Once near the bottom, plastic does not necessarily remain where it first lands. Bottom currents can resuscitate light pieces, slide them downslope, or bury them beneath later sediment. Trench walls provide gravitational pathways because earthquakes and sediment loading can trigger submarine landslides. These dense sediment flows, called turbidity currents, race downhill and deliver mixed material into deeper basins. Plastic already resting on shelves, slopes, or submarine canyons can be entrained within such flows. Direct samples from deep settings show that human debris can accompany sediment far beyond its original release point. However, finding fibers in a core demands strict contamination control from clothing, air, equipment, and laboratory handling. Visual identification alone can misclassify natural fibers, so chemical confirmation strengthens claims about polymer composition. Sediment cores reveal burial through layers, while remotely operated vehicles provide context for larger visible debris. These two methods answer different questions and cannot fully reconstruct every stage of transport. Trenches vary greatly in shape. Sediment supply, current strength, nearby population, and connection to canyons. Their trapping efficiency should therefore differ, making global generalizations from one site scientifically unsafe. The verified context is a network of pathways, not a single conveyor carrying every fragment downward. The mystery sharpens at the trench axis, where topography can concentrate particles from a much wider catchment. Imagine a steep-sided valley receiving repeated avalanches from both walls and slow fallout from the water column. Material reaching the narrow floor has less horizontal space, potentially raising local concentrations relative to adjacent planes. Weak circulation within enclosed depressions may lengthen residence time, though direct current records are limited in many trenches. Stronger currents can also focus debris by winnowing fine natural sediment and leaving certain particles behind. Thus, the same observed concentration might reflect enhanced delivery, reduced removal, or selective preservation. Burial protects plastic from sunlight and surface abrasion, but pressure and cold do not make every polymer permanent. Chemical changes, microbial colonization, and mechanical fragmentation can continue, often slowly and unevenly. Researchers must distinguish disappearance from fragmentation, because smaller particles may escape detection without being mineralized. If you see a bag on a hadal image, the observation establishes debris there, not its age or source. Printed markings may suggest origin, but circulation and reuse can separate manufacture, disposal, and final deposition by years. Microplastic abundance poses another challenge, because sampling volumes are tiny compared with the scale of a trench. Patchiness means neighboring cores may contain different counts after a single sediment flow or localized accumulation. Fibers can align with currents, lodge among grains, or concentrate where organic material creates sticky surfaces. Deposit-feeding animals ingest sediment while seeking nutrition, accidentally taking plastic particles with natural food. An amphipod detects carrion, approaches the odor plume, feeds, and may encounter plastic associated with the baited environment. Such behavior demonstrates an exposure route, but experimental bait stations can alter normal local conditions. Gut contents show ingestion at a particular moment, not necessarily tissue absorption or population-level harm. Laboratory studies may reveal inflammation or reduced feeding under controlled doses. Yet, environmental relevance depends on realistic exposure. Chemical additives and absorbed pollutants create additional concerns, although transfer and biological effects vary among compounds and species. Plastic surfaces can host microbes, but calling them unique ecosystems may exaggerate ordinary colonization of available hard substrate. The strongest explanation for trench accumulation combines downward transport, topographic focusing, episodic mass movement, and potential retention. No single mechanism operates equally for fibers, films, pellets, ropes, and heavily fouled objects. Alternative explanations must also be tested, including contamination, sampling bias, and proximity to local maritime activity. Deep expeditions often visit known depressions, so discovery effort itself can make trenches appear disproportionately contaminated. Comparisons with nearby abyssal plains and slopes are necessary before declaring any trench an exceptional sink. Repeated sampling after earthquakes could reveal whether mass flows deliver fresh pulses of modern debris. Until such records exist, the trap is a supported mechanism whose strength remains location-specific. Plastic on the trench floor intersects life because hadal animals depend upon material arriving from above. Scavengers follow chemical cues toward rare food falls, increasing contact with debris carried along the same pathways. Deposit feeders process mud, select digestible particles, and return rejected or ingested material to the sediment surface. Burrowing animals ventilate shallow layers, potentially moving small plastic particles vertically through otherwise quiet deposits. Predators consume exposed prey, creating another possible transfer step without proving biomagnification. Microbes colonize polymers and surrounding organic coatings, but complete degradation is uncommon for many conventional plastics. Sharp fragments may cause physical injury, while fibers can occupy digestive spaces, depending on organism and particle dimensions. Actual consequences at hadal depths remain difficult to quantify because baseline biology is poorly documented. Trenches also preserve signals of consumption far away, connecting inland choices and ocean circulation to inaccessible habitats. A fiber shed during washing can enter wastewater, escape treatment, travel through rivers, and eventually join offshore sediment transport. A lost fishing line may sink directly, snag terrain, abrade into pieces, and persist near benthic animals. These Q-action consequence chains differ, yet both end with material entering ecological processes that never evolved around synthetic polymers. For you, the distance between a household object and a trench may feel absolute, but currents erase that intuition. The human stake is not that every discarded item reaches hadal water, but that no depth guarantees isolation. Reducing leakage near sources is more practical than recovering scattered fragments beneath kilometers of water. Deep cleanup could disturb fragile sediment and organisms while retrieving only a minute fraction of dispersed particles. Evidence from trenches, therefore, strengthens prevention without requiring sensational claims about complete contamination everywhere. It also exposes environmental inequality, because waste can cross borders and settle beyond the communities that produced it. Fishing, shipping, rivers, and atmospheric fallout contribute in proportions that vary by region and particle type. Assigning responsibility requires source evidence, not inference, from a fragment's color or shape. The ecological question remains precise. How much plastic arrives? How long it remains? And what effects occur at real concentrations? Future work needs matched samples from trench axes, walls, neighboring plains, and the water column above. Time series sediment traps could measure descending particles and show when fibers or fragments accompany seasonal biological flux. Chemically verified polymer analysis should include procedural blanks that expose contamination introduced during collection and processing. Current meters near the bottom could identify circulation capable of retaining, redistributing, or exporting different particle classes. After major earthquakes, new cores could test whether turbidity currents create distinct layers enriched in recent plastic. Age indicators in surrounding sediment may constrain burial timing, although they rarely reveal a precise disposal event. Experiments with realistic particles could examine ingestion and egestion without imposing concentrations far above environmental levels. Population studies would need enough specimens to separate plastic effects from food limitation, pressure stress, and natural variability. Standardized methods across multiple trenches would show whether apparent hotspots reflect geography or incompatible counting techniques. Researchers must also report non-detections, because absence under stated detection limits constrains the scale of accumulation. Smaller nanoplastics remain especially difficult to measure reliably in complex sediment and biological tissue. Claims about them should follow validated recovery and identification, not assumptions that every missing fragment became nanoscale. When you return to the colored fragment in dark mud, its presence records transport, concentration, and human reach. The scientifically honest conclusion is that some trenches trap plastic efficiently, but their rates, inventories, and biological costs remain unfinished measurements.